All right, today we're going on an architectural tour, but we're not looking at a skyscraper or some ancient monument. No, we are going to deconstruct the single most important structure in the known universe, the molecules that carry the blueprint for life itself. So what you're looking at right here are the blueprints for literally every living thing, from a tiny virus all the way up to you and me. They store and they transmit all the information needed to build and operate a life form. So the big question is, how are these incredible structures actually put together? Well, to understand any great structure, you have to start with its most basic building block, right? For DNA and RNA, that fundamental piece is called a nucleotide. And you know, it's actually amazing how simple this is. Every single nucleotide, the basic unit of our genetic code, has just 3 parts. At the very heart of it, you've got a 5-carbon sugar. Then there's a phosphate group, which kind of acts like the connector piece. And finally, you have a nitrogenous base. This is the part that actually carries the genetic information. Okay, let's zoom right in on that sugar. On the left, you see ribose, which is for RNA, and on the right, deoxyribose for DNA. Now look closely, the only difference is at that 2' position. See it? RNA has an oxygen and a hydrogen, that's a hydroxyl group, while DNA is missing that oxygen. It just has the hydrogen. Now that might seem totally trivial, but trust me, this one tiny chemical change makes DNA way more stable. It's what makes it perfect for storing our genetic blueprint for a whole lifetime. Next up are the bases. These are the actual letters of the genetic alphabet, and they come in 2 flavors, 2 chemical families. You've got the purines, which are the bigger ones with 2 rings, and the smaller single-ringed pyrimidines. Now, both DNA and RNA use adenine, guanine, and cytosine, but here's the key difference you gotta remember. DNA uses thymine, or T, while RNA swaps it out for a very similar base called uracil, or U. So how do these 3 parts actually become one? Well, it's like a super precise assembly line. The base snaps onto the sugar's 1' carbon, and then the phosphate group locks onto the 5' carbon. And voila! The result is one complete nucleotide, an energy-packed unit ready to be snapped into a growing chain. Okay, so we have our building blocks. Now let's see how they link up, arm in arm, to form the strong, repeating backbone of a nucleic acid strand. This right here is the chemical magic that builds the chain. It's called a phosphodiester bond. Basically, the phosphate group of a new nucleotide forms a super strong covalent link to the 3' spot on the sugar of the nucleotide already in the chain. This just happens over and over again, creating this incredibly strong and stable sugar-phosphate backbone. And what's really cool is that this process creates a fundamental property, directionality. Because of the way these bonds form, every single strand has a clear beginning, a free 5' end, and a clear end, a free 3' end. It's like a one-way street. And by convention, we always read genetic information in this 5' to 3' direction. This polarity is absolutely critical for, well, pretty much everything the cell does with its DNA. We've built a single chain, but that's only half the story. DNA's true power, its iconic form, is the double helix. So let's see how that final masterpiece gets assembled. And there it is, the famous DNA double helix. It's 2 of those chains we just built twisting gracefully around each other. And notice the brilliant design here. The sugar-phosphate backbones are on the outside, facing the watery environment of the cell. And where are the bases, the actual information? They're tucked safely away on the inside, protected from any chemical damage. It's genius. Now, it looks incredibly complex, I know, but its entire assembly is governed by 2 surprisingly simple and elegant rules. Any ideas what they are? Okay, rule number 1. The strands are antiparallel. Think of it like a highway. They run in opposite directions. One goes 5' to 3' and the other runs 3' to 5'. And rule number 2 is complementary base pairing. The strands are held together by specific pairs. A always bonds with T, and G always bonds with C. That's it. It's a strict, non-negotiable rule. And this perfect pairing means that one strand is a flawless template for the other. But hold on, those hydrogen bonds holding the rungs of the ladder together, they aren't the whole story, not by a long shot. The flat bases also stack on top of each other, really neatly, kind of like a stack of pancakes. This base stacking, along with the fact that the bases really don't like water and get pushed into the core, provides a massive amount of stability. Some would even argue it's more important than the hydrogen bonds themselves. So, we've built the helix. We understand the rules. Now, let's ask the big question, why this specific architecture? Why is it so perfectly engineered for its job of storing the code of life? Hey, remember that tiny little difference between the sugars we talked about? Well, this is where it becomes a huge deal. That extra hydroxy group in RNA is actually pretty reactive. It can attack its own backbone, which makes RNA much more likely to break down. But DNA, since it lacks that group, has a backbone that is way more durable. It makes it the perfect molecule for storing precious genetic information safely for decades. Now, for all its stability, DNA can't be a locked-up vault. The information has to be accessible. So if you apply a little heat or change the pH, the two strands can be gently pulled apart, or denatured. Then, when conditions go back to normal, they can find their perfect partner and zip back together, or renature. This ability to unzip and rezip is absolutely fundamental for copying DNA and reading its code. And what's really fascinating is that we can actually watch this unzipping happen in real time in the lab. See, when the bases are all neatly stacked inside the helix, they absorb a certain amount of UV light. But as the DNA melts and the strands separate, those bases become exposed and their UV absorption just shoots up. This phenomenon, called the hypochromic effect, gives scientists a direct window into the structural integrity of the double helix. Pretty cool, huh? So let's just recap that entire architectural journey. We started with the simplest possible parts: a sugar, a phosphate, and a base. These assembled into nucleotides. Those nucleotides then link together to form a strong directional chain. And finally, guided by those two elegant rules of base pairing and stacking, two of those chains intertwine to create the blueprint of all life. It really is a masterclass in molecular engineering, isn't it? Just a few simple components, when assembled according to these precise chemical rules, give rise to a structure of immense stability and incredible informational capacity. A structure capable of encoding the entirety of life. Which, of course, brings us to one final huge question. If DNA is built to be this incredibly stable, protected library like a Fort Knox of information, how on earth does the cell ever manage to get inside, turn the pages, and actually read the tightly guarded code locked within?